Is it better now?
Yes, sir.
Okay. So what I was suggesting is, so today you're going to learn something about functional foods, which is a very popular topic. And I'm going to show you evidence-based functional foods that could help you to improve your resilience against chronic diseases. When I say chronic diseases, I refer to non-communicable diseases, and how this could sustain a healthy lifestyle of yours. So let me just start with few statistics. Chronic diseases, as I said, you all know, it includes every non-communicable diseases. It could be heart diseases, stroke, asthma, arthritis, every disease which we all are familiar with. That is a major cause for disability across the globe. It kills around 41 million people per year globally, which is equivalent to 74% of all deaths globally. So just to tell you, more than 50% of the world population has one or the other chronic diseases. Okay, and 75% of the death is caused by this disease condition. Now, 80% of all the deaths, which I suggested here, 74%, among this 74%, 80% is caused by these top 4 diseases, which is cardiovascular disease, cancer, chronic respiratory diseases, which could be asthma, COPD, particularly COPD, and then diabetes associated problems, like it could be a renal failure or increased risk for heart disease and all this. So these are the major chronic diseases that is responsible for highest burden of disabilities as well as deaths. Now, what is more, if you look at the distribution pattern of these diseases across the age, you will see that the largest percentage of disease burden is between 15 to 49 years. And this is very worrisome. Why? Because this is the age of high productivity, economic growth. And at this age group, or in this age group, if people become sick, it not only threatens their existence because of the economic burden, it also can threaten our national security. So it becomes a very big responsibility on us as a life science scientist, that we develop some methods or some strategies to prevent or slow down non-communicable diseases among this particular group particularly. Yes, it is applicable to every disease, every age pattern, but this group is very, very important. Now, what are the risk factors that is responsible for these non-communicable diseases? Particularly, I'm referring to these 4 diseases: cardiovascular, cancer, respiratory disease, and diabetes. And these are the risk factors: smoking, air pollution, alcohol, physical inactivity or sedentary life, consumption of high ultra-processed food or high-calorie food or sugary foods, whatever we are used to eat as a snacks. It could be a Coke, it could be a juice. If somebody has looked at the nutritional fact of any Coke or Pepsi, you will see that 35 grams of sugar is there in 100 ml of your in your drink. So that's a very, very high. So chronic exposure to these conditions predisposes to certain metabolic factors or metabolic disorders, which could be hypertension, overweight, obesity, hyperlipidemia, and hyperglycemia. And these conditions predisposes to all these 4 conditions, 4 diseases, all the CVD, cancer, chronic diseases, and diabetes. Okay. What is important is, among these risk factors, except the air pollution, rest of the factors is under your control. It's up to you whether to select that particular or I don't know, choose that particular risk factor or behavior, and you can control the risk for the diseases. It could be smoking, it could be alcohol, it could be your physical exercise, how you do, and limiting your ultra-processed food. So it's all up to your control how to prevent these chronic diseases. Now, how chronic disease pathogenesis occurs. So in a very layman language, I'm going to tell you, when you get exposed to risk factors, which I just explained in the previous slide, the first trigger is oxidative stress. It causes oxidative stress. This oxidative stress leads to inflammation. And inflammation then feeds back to oxidative stress. So these two conditions, these two events happens hand to hand. And with days of exposure, with the days of increased oxidative stress inflammation, you'll see there is an increase in cell death, or there is an increase abnormal cells, or it could be dysregulated immune response. A dysregulated immune response could be that it could be somebody could be having an immunosuppressive phenotype, or somebody could be having a hyper immune activation step. Both could be possible. And with time, you'll see that there'll be a tissue injury and impaired tissue repair leading to disease condition. It could be any of these diseases. Each of the organ, if it is in cardiovascular, we call vascular, dysfunction happens, vascular disease happens, you have atherosclerosis. In cancer, abnormal cell, a rogue cell becomes an uncontrolled proliferation. In chronic respiratory diseases like asthma and COPD, you lose tissue itself. There is a loss of tissue, or there is airway remodeling that causes obstruction of your airflow. In diabetes, you lose beta pancreatic cells, and then because of increased oxidative stress, inflammation that causes renal failure, retinopathy, nephropathy, all kind of morbidities are associated with diabetic conditions. Now, just to be on the same page, what is oxidative stress? Oxidative stress is a state where you have excess accumulation of free radicals because of inadequate antioxidants. Okay, I want you to be clear with the definition. Excess accumulation of free radicals, and free radicals could be a reactive oxygen species, which could be a superoxide, or it could be a hydroxyl radical, or peroxide nitride, or hydrogen peroxide. All these are free radicals. Or it could be electrophile. So when you have increased levels of this and you have reduced levels of antioxidants, there is a state created which we call oxidative stress. And what are antioxidants? Antioxidants are the one which quench these free radicals. They can react one-to-one. If they react one-to-one and neutralize the free radical, we call them as direct antioxidants. And there are enzymes which call them indirect antioxidants, because they work in such a manner that they use a cofactor to reduce these free radicals. So you have both direct and indirect oxidants. However, whenever you have low levels of antioxidants in your body, or if the cells are having low levels of antioxidants and increased free radicals because of exposure to environmental risk factors, it could be a smoke, air pollution, high calorie diet. These increased reactive oxygen species or free radicals will cause damage to your biomolecules, which could be a lipid, which basically the membrane lipids, which are phospholipids, or it could damage your lipoproteins, which are LDL and HDL. It can damage your proteins, leading to its aggregation or fragmentation, inhibition of enzyme activity. A certain modified proteins can become pro-inflammatory in nature, and DNA damage can also occur, that leading to a mutation or epigenetic changes that could lead to cancer. So this is a general pathway how disease happens. Now, how does ROS or oxidative stress activate inflammation or induces inflammatory signaling process? When there is excess of reactive oxygen species like superoxide, hydroxyl radicals, or hydrogen peroxide, there are certain sensors in your cells that will sense these free radicals and will get activated. At the end of the day, these sensors will activate certain transcription factors. One transcription factor, which we call is NFKB, and there is also called as AP1. And there are certain MAP kinases also, which could be ERK, P38 junk. Once they get activated, they go into the nucleus and they bind to certain elements, or we call motif element, to specific motif elements, and they activate the gene expression of cytokines. You all know what is cytokines and chemokines. Cytokines are the pro-inflammatory in the sense they recruit more inflammatory cells. Chemokines helps to recruitment also. And these cytokines and chemokines, they can act in a autocrine manner or a paracrine manner, and they help to recruit inflammatory cells, which are neutrophils, macrophages, lymphocytes. For example, if any of us get an infection, it could be bacterial or virus, within few minutes, you'll see increased levels of cytokines. Within half an hour, you see loads of cytokines and chemokines, and these guys will recruit these macrophages and neutrophils and lymphocytes into the lungs. And these, the function of these inflammatory cells is to kill the bacteria or virus. However, when they come, they also release a lot of free radicals. They also release a lot of proteases. So these proteases and free radicals can induce tissue injury and cause cell death. And with time, you'll see that there is excess of tissue inflammation and injury which cannot be repaired, so that leads to a disease condition. And just for a good example would be COVID, which we have experienced a couple of years back. In the COVID virus, caused inflammation in the lungs, and the patients were having high levels of these immune cells, leading to a lot of damage, leading to fibrosis, and cell death, leading to lung failure. Now, as a good indicator of severity of the disease, people were using IL-6 as the indicator of severity of the disease. So just to tell you, so if I want to measure inflammation, I can count the number of these inflammatory cells at the lesion or in the lung itself. So please remember that. I'm going to talk about this later as we go on. Okay. So this is how, in a simplistic way, how oxidative stress can induce inflammation. Just going back to the same process. Okay. So you have risk factors. It could be your smoking. It could be anything, or even pathogen. It can activate oxidative stress. Oxidative stress will lead to inflammation. Inflammation, again, I defined you as recruitment of more inflammatory cells to the place of where there is an injury or infection. These inflammatory cells, they produce reactive oxygen species. Why? Because of activation of NADPH oxidases. And this together leads to all these processes. Okay. So technically, if I can prevent oxidative stress or inflammation, I can prevent these diseases. And you all are very familiar with anti-inflammatory medications like corticosteroids or even non-steroid anti-inflammatory drugs. They work on reducing inflammation. And you also are familiar with vitamin E, vitamin C. They are supposed to work against oxidative stress, but the data is very, very big. So as we go on, we're going to talk about that. So with that little background, if I want to, if I plot the relationship between oxidative stress and the disease progression, because remember, the disease progression happens over the years, most of the non-communicable diseases. It could be diabetes, it could be cardiovascular disease. It takes a minimum 10 to 15 years to develop a clinical sign or clinical symptom of a disease, right? Now, if I plot oxidative stress against the disease progression, this is how a sigmoidal curve looks like. So you'll have an initiation stage, you will have a subclinical stage where you'll have no symptoms, and then you have a clinical stage where you have symptoms. Now, during the initiation stage, you have very low levels of oxidative stress, but during the subclinical stage, the rate of increase is very, very high. And during the clinical stage, you are associated with highest level of oxidative stress. Now, if I use an antioxidant therapy, remember what is antioxidant? Antioxidant is the one which will reduce the levels of free radicals or reactive species, both when it is reduced. If it is given during the initiation stage, you can prevent or inhibit the disease progression. Correct? Now, if you take antioxidants during this stage, subclinical stage, probably you can halt, even reverse or delay the process of the disease progression. But when you take during the clinical stage, when the disease has already happened, very likelihood that you may have a minimal effect. It may slow down, or it may not have any effect. Now, what is the evidence for this? Now, you all have heard that, and you have been prescribed during different disease condition, vitamin E, vitamin C, beta carotene. These trials have been done multiple times, but the impact of these vitamin E, vitamin C has been very, very inconsistent and minimal. Most of the clinical trials have shown neutral effect. There's no effect of vitamin E, vitamin C. And the reason could be multiple. One reason could be that you are treating, or rather you're taking this vitamin E, vitamin C during this stage, clinical stage. When you take that clinical stage, already disease has opened, has happened, and there is a maximum tissue injury and all that. At that particular stage, the effect of vitamin E, vitamin C, which are quenchers, they work one-to-one. That means once a free radical reacts with vitamin E is no more functional. It becomes inactivated. Okay, so you need to keep on replenishing this vitamin E, vitamin E for function to happen. So again, the bioavailability of these vitamins at the tissue injury or at the disease tissue is very, very minimal or questionable whether it can reach to that stage. However, what people have shown that if you have combination of antioxidant, the effect is much more. Because, for example, if I take vitamin E or vitamin C, they work together in such a way that once vitamin E get oxidized by free radical, vitamin C can go reduce the vitamin E and brings back its in a reduced state. So this way, a cycle is created where it come in a, the vitamin E is active for a longer period of time. But it depends. There are small studies which have taken vitamin E, vitamin C during this stage. People are shown yes, it can protect it, but at this stage, very minimal effect. But it's not that there are several epidemiological studies suggesting that any diet rich in vegetables and fruits protect from diseases. Another, the reason why these clinical trials with vitamin E, vitamin C failed is that often these trials are very for a short period of time, 6 months to 1 year or 2 years. But this disease process happens over a period of at least 5 to 10 or 15 years. So you need to take a longer time. So in contrast, the multiple epidemiological studies, you're not talking about few hundred, I'm talking about thousands of people were recruited in this epidemiological studies, and they found that people who consumed more of vegetables and fruits, they were protected from chronic diseases. It could be heart disease, it could be diabetes, even cancer. Okay, so there is a truth that if you enrich your diet with vegetables and fruits, it can protect it. And you all know that nowadays our diet is free or minimal amount of vegetable fruits are there, particularly for this generation. When probably few of you are, I'm already 50 years old, and when I was growing, it was not, I would say, compared to present generation is very easy to become diabetic or cardiovascular disease. Why? Because the access to calorie-rich food is very high. I step out of my home, I have a bakery, I have I have all kind of restaurants, right? But during my childhood or during my youth period, I didn't have this so close access to these kind of what you call nutrient-free diet resources. Plus, I had less access even with financially also. But today's generation has both, and therefore it's very easy to become sick at a very early age because of the access to these particular unhealthy food products. Now, one more thing I want to introduce it. You can see that during the initiation stage, though you're like, for example, if somebody is a smoker, and if that person smokes every day 1 pack cigarette for minimum 10 years, he develops COPD. But there are patients or there are subjects who, despite smoking for that many years, they don't develop any disease. Okay, so that's first thing. And you see that oxidative stress is minimal during the initiation stage compared to your subclinical stage. So this tells you that there are intrinsic defense mechanisms in your body that defends against this risk factors. It could be any kind of risk factor that produces oxidative stress. Okay, I'm going to introduce to one particular factor that is responsible for regulating or upregulating all the antioxidant defenses in your body. That particular factor is called as NRF2. It's a protein. It's a transcription factor. You all recall that transcription factors are the proteins that can go and activate transcription and regulate not one, multiple genes. So this transcription factor was discovered late in the 90s, and later it was found that is a major regulator of nearly all cellular antioxidant defenses. I'm not talking about vitamin E, vitamin C. I'm talking about antioxidant enzymes as well as antioxidant peptides. Okay, to tell you a good example. So NRF2 regulates genes or other proteins that help in synthesizing glutathione levels. Glutathione is a very powerful antioxidant present in millimolar concentration in your blood. So the enzymes responsible. So JSS is a 3 amino acid peptide, glycine, cysteine, and glutamine. So all these proteins or other enzymes responsible for synthesizing glutathione is regulated by NRF2. Now, glutathione is a very good cofactor for this enzyme glutathione peroxidase. Glutathione peroxidase helps to reduce the peroxides to alcoholic form or non-toxic form using glutathione. If glutathione is not there, GPX will not work. So now, in the process, what happens? The glutathione will be oxidized to oxidized glutathione, and the substrate will be reduced to water or alcoholic form. And this oxidized glutathione is recycled back to glutathione by this enzyme called glutathione reductase. And this enzyme is also regulated by NRF2. And then glutathione reductase, for its enzymatic action, it needs NADPH. So there needs to be a constant repletion of NADPH. And NRF2 regulates even glutathione 6-phosphate dehydrogenase, which is a major synthesizer of NADPH. So just by activating NRF2, I can get a coordinated induction of multiple cellular antioxidants which work together. It's not like one antioxidant, it's multiple antioxidants. They work in a network fashion. So the effect or efficiency of resisting oxidative is powerful compared to if you take a vitamin E or vitamin C. Okay, so like that, you have peroxyredoxin, which is also a peroxide reducing enzyme. You have heme oxidase, which produces bilirubin. Bilirubin is a very powerful antioxidant, better than glutathione levels. So, and then you have phase 2 enzymes. One of them is NQ1. What are phase 2 enzymes? Phase 2 enzymes are metabolic enzymes that can biotransform a xenobiotic into a non-toxic and form, which also helps to excrete it, eliminate from your body. So NQ1 is one of them. So NQ1 is a surrogate marker of NRF2 activation. So because I told you NRF2 is a transcription factor. So instead of measuring NRF2 as an indicator of NRF2 activity or NRF2 induction, we measure the gene targets, which could be NQ1, GCLC, GCLM, and hypoxia. These are the potential or surrogate targets of NRF2. So we measure this as a surrogate indicator of NRF2 activity. So I want you to remember this enzyme NQ1, GCLC, GCLM, and glutathione for the future. As we go on, you're going to hear me more about that. Now, how does it NRF2 work in your cells? So NRF2 is switched off in a normal cell. Very minimal activation is there. It is switched off because it is held by two protein, a protein called KEAP1. Two molecules of KEAP1 hold the NRF2 in the cytoplasm and link it to the cytokine filaments. Okay, and this KEAP1 acts like an adapter molecule to this particular complex called CUL3 complex. So the function of CUL3 is to ubiquitate NRF2 and promotes this degradation. So constantly, KEAP1 will act like a bridge between CUL3 and NRF2 and promotes NRF2 degradation. So in a normal cell or unstressed cell, NRF2 levels is very, very less. However, in response to any kind of environmental stressors, it could be, I told you smoking, air pollution, whatever. Whenever there's a small change in the redox status of cell, that when I say redox means there is increased ROS levels, the KEAP1 is a cysteine-rich protein that gets modified and it's no more able to bind or hold on to NRF2. So NRF2 is free in the cytoplasm and it goes to nucleus and binds to DNA. Binds to a DNA element or basically a 15 nucleotide base pair cis element, which we call as ARE element, and then activates the transcription of the gene. Now, this ARE is, wherever this ARE is present, in the sense, whichever gene has this ARE in their promoter gene, all those genes will be activated. And by doing genomic sequencing and microarray approach, we have found that NRF2 can regulate more than 100 genes. Most of them, they function as a cytoprotective defenses. So just by activating NRF2, we can increase hundreds of cytoprotective genes, which can help to resist any oxidative stress or inflammation, which is caused by the risk factors. So I hope you're getting the point. So once you have increase of these defense molecules, you will have reduced oxidative stress, inflammation, cell death is less, and therefore you protect from the disease. Now, over a period of time, so when cells have this very powerful machinery, why is that we get disease? What we have found is, and we and other people have found it, over a period of time, somewhere because of some defect, the NRF2 activity goes down. That means there is a suboptimal activation of NRF2. As a result, all these protective enzymes and proteins are not able to be synthesized. As a result, you get more disease. Okay, how do we know that? Now, to understand the importance of this protein NRF2, we have made NRF2 knockout mice, wherein we have knocked out the gene of NRF2. As a result, this particular mouse will not have a functional NRF2 gene. So if you don't have functional NRF2 gene, what happens? There will be no induction of all the antioxidant defenses that I said in the last slide. So when we compared a normal mice with NRF2 knockout mice and stressed with any kind of risk factor, it could be tobacco smoke, air pollution, high-fat diet, microbes, radiation, the knockout mice experiences and accumulates more damage. It could be oxidative stress damage, it could be inflammation, it could be tissue damage. Because of excess of these, even the disease severity is very, very high in these knockout mice. Okay, compared to the wild type mice. Wild type also will get it, but not to the extent of knockout mice. And what kind of disease we are talking about? Every disease in your body caused by these risk factors, which drives oxidative, we and other people have shown that knockout mice is very sensitive for these diseases. Knockout mice is healthy in the normal conditions until 1 year. After 1 year, it develops autoimmune disease, but till 1 year, mouse is fine, as good as the wild type. But if you stress the mice shows severe phenotypes. Okay, and now to the knockout mice, if I feed this knockout mice or treat this knockout mice with an antioxidant, the knockout mice shows less degree of injury and less degree of disease. Thus tells you that antioxidants can help to reduce the severity of the disease or initiation of disease. But the important point what I want to make is, now if I have an NRF2 inducer or pharmacological activator for NRF2, and if I treat the wild type mice, it can protect the wild type mice from all the injuries or basically oxidative stress, inflammation, tissue damage, and therefore protect from disease. Okay. In most of the cases, we have shown that a pharmacological activator will prevent or mitigate the disease progression. Any disease, it could be heart disease, it could be COPD, it could be obesity also, because NRF2 is shown to regulate even metabolism also. It regulates it can reduce even arthritis also. In mouse models, people are shown. And because of this market value of NRF2 pharmacological activators, many companies are behind developing drugs for NRF2, targeting NRF2. And 2 drugs are already been approved by the FDA. One is dimethyl fumarate, which is sold as Tezspire for treatment of multiple sclerosis, which is an autoimmune disease. And there is one more called bardoxolone, which is being recommended or prescribed for treatment of pulmonary hypertension. Okay, so this is what the market value of NRF2. So by increasing NRF2 activity, you can increase all the cellular antioxidant defenses, which will prevent inflammation, cell death, immune activation, and therefore it protects from the disease. That's the whole concept I want you to buy at this stage of the presentation. Now, what is interesting is a lot of fruits and vegetables, they do contain a lot of phytochemicals, which are very potent activators of NRF2. Okay, so now, what are these? These are certain polyphenols. Most importantly, I'm going to talk about isothiocyanate compound, which mainly sulforaphane and monogenine. And how we and other people have shown that this could be, by consuming this sulforaphane and monogenine, it can reduce disease progression as well as help in disease prevention also. Okay, now coming to the definition of functional foods. Okay, now a definition of functional foods, just by looking at the terminology, functional foods means foods beyond nutrition. That means foods that have function beyond just giving you calories. That's a simple definition. But in literature, you will see functional foods definition has been a little more confusing. There's less differences between healthy foods and functional foods or superfoods. Okay, like whole grains can be called functional food or a healthy food. It's still, there's a gray line between this. So to be on the same page, functional foods, we can refer them as foods that are formulated or designed, okay, appears to be similar to conventional foods, but contain substances or living organisms, which is basically bacteria, which have possible health enhancing or disease preventing value at a concentration that is both safe and sufficiently high to achieve intended benefits. Okay, so it's a formulated or designed foods. That means you're enriching the food with a particular substance or chemical or phytochemical. It could be a fortified orange juice with the calcium or any other omega-3 fatty acid. That could be called a functional food. So you're formulating it rather than just taking a natural food. Like, for example, broccoli, we know it's good. We know garlic is also good. But we cannot call this as a functional food because we need to make it in such a way that it is easy for consumption as well as has health benefits to that. Okay. So just to be on the same page, I want you to remember that it's a formulated design of functional foods. Now, what are nutraceuticals? Nutraceuticals are health-promoting, nutrient-rich dietary products that are derived or extracted from food sources, but supplied as a pharmaceutical preparation in the form of powder or pill or liquid. Okay, so it's a kind of a extracted nutrient or phytochemical from a food source, and it is being supplied in the form of powder or pill or a liquid. So that's the difference between nutraceutical. So this doesn't have any calorie value, but this has a calorie value. At the same time, it has certain health promoting chemicals also. Okay, clear? Just can you confirm that you're able to hear me and see the slides? I'm sorry, I totally forgot to ask.
Yes, sir. Yes, sir.
Yes, sir. Yes, sir. Visible.
Thank you. So I hope I'm clear with the definition of functional foods. Now, I'm going to give a few examples, okay? And so that you understand and appreciate the value of functional food. The first food I'm going to talk about is a broccoli sprout containing sulforaphane. Sulforaphane is an isothiocyanate compound present in broccoli, okay? Now, this was discovered by, rather kind of identified by, Dr. Paul Talale. So I was at Hopkins, and during that time I had an opportunity to interact with him. So he passed away a few years back, but very, very highly accomplished scientist he was. So he was the one who proposed chemo prevention or chemoprotection of cancer. So he was interested in preventing cancer by increasing certain phase 2 enzymes that will metabolize carcinogens and eliminate from your body. Clear? So chemoprotection from cancer. So you go to use certain chemicals that will increase these enzymes in your body, and these enzymes are responsible for detoxifying carcinogens. It could be carcinogen, it could be from a tobacco smoke or from an air pollutant, or it could be from a diet, or it could be aflatoxin, whatever it is. They are metabolized by set of phase 2 enzymes. One of the enzyme is NQ1. The other enzyme is GSTs, UGT. There are many are there. So one of the enzyme is NQ1, and he was working on this enzyme NQ1. And he developed this NQ1 bioassay to screen plants or food plants and identify which food plant has the highest level of NQ1 inducing activity. Okay, when he was working, NRF2 was not discovered. Later, it was found that NRF2 is the one I referred as is the key regulator of NQ1. That means NRF2 regulates NQ1. Okay, so if NRF2 is activated, NQ1 will also increase. And not only NQ1, there will be other GST, glutathione synthesizing, all will be upregulated. So later we came to know that NRF2 regulates NQ1, and this became NQ1 bioassay became a bioassay to screen for NRF2 activators also. But when he was working, he developed this NQ1 bioassay just to identify plant foods that can increase detoxification of carcinogens. And NQ1 was his target enzyme. So what he did is he asked his student to go to the market and get all the vegetables. And later he found that the certain Brassica family vegetables were having high NQ1 inducing activity. And he then screened more Brassica family vegetables and found that broccoli is having highest level of NQ1 bioactivity. Okay, and he found that this bioactivity is because of this compound called sulforaphane, which is an isothiocyanate compound, NCS. It has a sulfur-containing compound, it is. But sulforaphane is present in its precursor form called glucophanin. So glucophanin is a secondary metabolite. which needs to be converted to sulforaphane, and it is converted by the enzyme called myrosinase enzyme. So myrosinase enzyme, as you all know, it is released when you crush the vegetable, when you crush the plant cell. Okay, so when it is crushed, the glucophan will convert to sulforaphane, and since sulforaphane is an isothiocyanate compound, it has a very pungent smell, and it repels the predators from eating the crop. So that's one of the function for the plants. But what Dr. Talale found is sulforaphane is a very good inducer of NQ1, and later we found that sulforaphane is a very good activator of NRF2 also. It's a very potent NRF2 activator. So now, when you crush myrosinase release, even your gut microbiota contains myrosinase enzyme. It releases myrosinase enzyme. So gut microbiota can also digest or rather convert the glucophan into sulforaphane. So now what he did is he screened every part of the broccoli and found that seeds have the highest content of glucophan. But seeds are very bitter. So he made seeds into sprouts. And he found the sprout contains the highest level of sulforaphane or rather glucophan. It is almost like, you can see 3 grams of broccoli sprout is equivalent to 150 grams of broccoli floret. So that much content is there in the broccoli sprouts. Almost like 50 times more glucophan is there in the sprouts rather than the floor. So now he basically screened different broccoli seeds variety and identified one broccoli seed that has the highest amount of glucophan, and he patented these broccoli seeds and broccoli sprouts from that. And now if you go to US, you will see that broccoli sprouts are sold in supermarket, and you will see it is patented from Johns Hopkins. So he has done that also, and translated to the community also. Now he also he came to know that the myrosinase is a key determinant of conversion of glucophan into sulforaphane. But if you eat the sprouts, there is a differential conversion of glucophan into sulfur. That means every individual may not convert all glucophan into sulforaphane, because that is determined by gut microbiota as well as how much he chews. A lot of things are there which will affect the conversion of glucophan into sulfur. So what he did is he developed a broccoli sprout extract rich in sulforaphane. What did he do? He took the sprouts and boiled it in the water. Please remember this, okay? He boiled into the water and filtered it. So water extract contains rich in glucophan. Now so what it is, he freeze-dried this glucophan powder, rather this extract into a powder, and he called it broccoli sprout extract powder. So it's rich in glucophan. So you can measure whatever amount you want to give of glucophan, and people can consume that. Then he also did one more thing, just to prevent differential conversion of glucophan to sulforaphane. He treated glucophan with myrosinase enzyme, and he converted the glucophan completely to sulforaphane. So now he has a sulforaphane enriched broccoli sprout extract powder, which this powder can be added to a juice, or it can be made into capsules, and people are taking it. And I'll show you some data how it is showing good effect, beneficial effects. Okay, so important point is he has converted the glucophan into sulforaphane by using myrosinase enzyme. I want you to remember this process as we go on. Okay, now just to show you a few studies. Now this is a very good study published in Science Translational, a very high impact journal, where they found that by giving capsules either with sulforaphane or dosulforaphane or placebo, the diabetic to obese patients with uncontrolled diabetes condition, they found that people who consumed the broccoli sprout extract rich with sulforaphane had a better control of blood glucose. And mechanistically, they found that sulforaphane down-regulated expression of certain genes involved in the gluconeogenesis, right? So that was the mechanism, and it was significantly shown to be different between the placebo and the treatment group. That's the first study. This is another study, my favorite study. What they did is, this study was done in China, in a high polluted city, Chidang. And there, I told you that sulforaphane is an activator of NRF2, and if you activate NRF2 will increase phase 2 enzymes, which are NQ1, GST, UGT, and they are the one which helps to detoxify and eliminate any carcinogen or xenobiotic like air pollutant chemicals also. So what they did is they had they recruited healthy subjects from that community, and they gave broccoli beverage or a placebo. Broccoli beverage contained 360 grams of glucophanin rich and 24.8 grams of sulforaphane rich powders dissolved in water and pineapple juice. So pineapple juice used to mask the smell and all that. Pineapple and lime juice with a ratio of these ones. The placebo contained the same liquid, but no glucophanin and no sulforaphane. And what were they looking at? They were looking at urinary metabolite of air pollutants. What air pollutants? Benzene, acrolein, and crotonaldehyde. These three chemicals are metabolized in your body and excreted in the urine in the form of glutathione conjugates. So they are looking at the glutathione conjugates of benzene, acrolein, metanol, which we call mercaptric acid derivatives. Now, the moment they took this beverage, from day one, they could see there is an increased excretion of these air pollutants from the urine of people who took the beverage compared to the placebo. So green one tells you the increase in the excretion of this benzene, whereas the placebo. So this is very, very interesting. That means every day if you take this beverage, you are able to excrete these carcinogens or these bad chemicals from your body, and therefore the accumulation of these bad chemicals will be less in your body, and there will be less likelihood of oxidative stress, inflammation, cancer, and so on and so forth. So that's the whole concept here. Okay, next one. This is a study where they were studied among the kids with autism. Now, NRF2, when it's get activated, I told you it regulates more than 100 genes and proteins. One of the targets of NRF2 is heat shock proteins. And there are a few heat shock proteins NRF2 can increase also. And these heat shock proteins can be very helpful in autistic syndrome. So what they did is they took kids who are autistic, divided them into two groups, placebo and sulforaphane, basically capsule. And they supplemented the kids with these tablets, and they found that the moment the kid take sulforaphane capsule, his score, aberrant behavioral scores, which includes irritability, lethargy, stereotype, hyperactivity, was significantly reduced among the sulforaphane group compared to the placebo group. You all know autism behaviors. And there is a spectrum of behavior. So it's not one particular condition, it's a spectrum of disorder. And these are the common behavioral abnormalities. They are very irritability, lethargy, stereotype, hyperactivity. And what was, I was also surprised to see these results. And the moment they take, it was reduced. Now, what is interesting is they stopped taking after 18 weeks. The moment they stopped, did you see that phenotype is coming back? So as and when they were on the particular sulforaphane dosage, they could reduce the symptoms, but they stopped it, it came back. So again, this has been shown to be very effective in this condition also. So like this, there are many studies people have shown that sulforaphane enriched broccoli sprout extract can prevent cancer, can prevent viral diseases, basically like even the flu virus and the symptoms of flu virus. It's been shown to be reduced, and it's been used even preventing the radiation with dermatitis. So there are many trials. So this is just a glimpse of how a functional food or an NRF2 inducer can help to prevent the disease state. Now, coming to the next group, Moringa oleifera. When I came back to India and joined JSS, my first project I wanted to do was to screen food plants which have potential to activate NRF2, because I had worked on broccoli when I was in Hopkins. So I know how impactful broccoli, so NRF2 activating chemicals could be in the food. So consciously, I wanted to screen food and medicinal plants for potential NRF2 activity. And I gave this project to two of my students, PhD students, and they screened many plant, food as medicinal plants. And they identified two plants having a good NRF2 activity. One was piper beetle, and the other one is Moringa oleifera, which is the Nuggei soup. Okay, so I'm going to show you the results from Moringa oleifera. And to tell you more about this particular compound, Moringa leaves contain this secondary metabolite called glucomoringin. And this glucomoringin is converted to moringin by this enzyme myrosinase, same enzyme responsible for converting glucophan into sulforaphane. And moringin is also isothiocyanate compound like sulforaphane. So this was interesting. When we first found that NRF2 moringa leaves can activate NRF2. This is a water extract of we did a water extract of these leaves, water as well as organic extracts. And what I'm showing is a water extract of moringa leaves. And it showed good activity. And when we went back to literature and found that, yes, moringa leaves contains the glucomoringin, which is converted to moringin, which is isothiocyanate. Same way as your glucophan is converted to sulforaphane, by myself, same thing happens here. Okay, but at home, how do we cook moringa leaves? So moringa leaves, so you all know broccoli is not that commonly found in India. It is more a high-end recipe during weddings. But as moringa leaves is common, it's a poor man's broccoli, I can say, right? It's available in the market. And we used to eat a lot, but these days it has come down. But if you look at how we cook the moringa leaves at home, what we do is we cook the leaves, we take the leaves, boil it, and then either we use the water or only the boiled leaf, the tender leaf, or the both, right? And then we make whatever we need. We can make a sambar, we can make a soup, whatever we want to make, we make it. But what happens is when you are boiling the leaf, it kills the myrosinase enzyme. And therefore, the conversion of glucomoringin to moringin is limited. There's nothing, no conversion is there. So we thought that how about we make a food preparation or process such a way that it can increase the moringin content. So we did a simple tweaking of this cooking process. We mimicked every cooking process that we adopt at home. And the last one, MED, what we did is we grinded the leaves before boiling. Take the fresh leaves, grinded it, and either we filter it, and the filtrate was freeze-dried and used for testing. But here, we grinded it, incubated for 15 minutes, or 15 to 20 minutes rather. You can keep it even for half an hour also. After 15 to 20 minutes, we boiled it to make it palatable, filtered it, and this filtrate was made into freeze-dried powder, and we call this MED. Okay, here we did immediately boiling, boiled it, and then we did the same process. Either you take the filtrate or grind it, take the leaves. So we have MEA, B, C, D. Okay, and now we looked at NRF2 activity by looking at the same NQ1 bioassay that Dr. Talale had used it. And we found that, I am not showing the data here, among all this formulation, MED was the most potent activator of NQ1. So this addition of this step, wherein we incubated for 20 minutes, was sufficient to convert glucomoringin to moringin, and that increased the activity of NQ1 activity in the formulation. Also remember that besides the moringin, moringa leaf contains many polyphenols, and polyphenols are also good activators for NRF2. It also contains high levels of calcium and maybe small amount of iron and many other minerals. So it's a super rich functional food, right? So now we tested this MED formulation against different, we basically looked at different bioactivity. This is bioactivity showing that it can increase NQ1 enzyme activity in a very dose-dependent manner. And even at high as a 500 microgram per ml, it's not toxic, and it is a very good inducer of NQ1. Then I told you that GSH glutathione is very important antioxidant, and we showed that, yes, it can increase glutathione levels in the cells, and the potency to induce glutathione was equal to sulforaphane, almost equal to sulforaphane. Then we looked at the antioxidant activity of cooked moringa leaf water extract, and we found that by pretreating cells with moringa extract with MED formulation, it can protect from cell death induced by different pro-oxidants. It could be AAP, which is a free radical generator, or a tertiary butyl hydroperoxide, or even arsenic. Different mechanism how it causes cell death. In all these toxicants, we found that by pre-treating the cells with MED, it protected from cell death. So it's a very good antioxidant, and it can protect from cell death caused by oxidative stress. Next, we wanted to see if it can increase NRF2 activation or NR2-regulated antioxidant genes in mouse in different tissues. So we basically fed orally this powder dissolved in water, and then we isolated the small intestine, lung, and liver, and then we measured the NRF2-regulated antioxidant genes, which are NQ1, HO1, GCLC. In all the tissues, we found there was a significant increase in these antioxidant defenses. Next, we wanted to test the therapeutic effect of this moringa extract. So what we did is I worked on air pollution also. So we exposed the mouse to air pollution. And you all know air pollution causes lung inflammation. In the mouse, we can measure the lung inflammation by counting the total number of cells accumulated in the lungs. So this is a vehicle. When we expose it to air pollution, the number of total cells in the lung increases. Basically, we wash the lungs, and in the lungs, all the inflammatory cells comes into that lavage. It could be macrophages, it could be neutrophils, lymphocytes, and then we count it to see how much is inflammation. Now, the mouse, when they were exposed to air pollutant, as well as given moringa leaf extract, we could see a significant decrease in the inflammatory cell numbers, suggesting that by taking this moringa extract formulation, you can protect yourself from air pollution associated lung inflammation. Okay, this is a data which has been published last year. Now, how do I translate to the community? How can we make such a way that this can be used by everyone in the community? For that, we did a simple experiment. We took the moringa leaves, washed it, grind it, incubated 15 minutes, and then boiled it, and then only collected the soup. Okay, only the extract part, hot soup. And then we wanted to see if this moringa soup contains the activity. Yes, it contains activity. At different concentration, we found that it can induce NQ1 in the cells, and it also reduces inflammation. What I'm showing you is the level of nitrate levels accumulated in the media after the cells were exposed to lipopolysaccharide, which is a ligand for TLR4 ligand. It's LPS is a component of the bacteria. So it has both antioxidant as well as anti-inflammatory properties. So now, this soup we are trying to test in the in patients to see if it can reduce inflammation, it can reduce oxidations, and it can prevent the disease phenotype. At least we are working on the grant where we are targeting COPD population, and COPD, it's caused by tobacco smoking and air pollution. So we are trying to test if this can reduce the symptoms in these patients. So maybe next 2 years, I can tell you if at all the grant get funded, I can do this trial and tell you how this works. So that is about the second product. Now I'm going to switch gears and tell you a little bit about gut microbiota and foods rich in fiber and fermented foods. Okay. You all know by now that your gut contains 100 trillion bacteria, okay, which is 10 times more than the total number of cells in your body. Your body approximately is made up of 10 trillion cells. It's at a higher one only, but your gut contains 100 trillion cells. That means 10 times more than your cells. So that itself is an organ. So what are these gut microbiota doing? They are digesting the food which was undigestible by your intestinal enzymes, which we call dietary fiber. So dietary fiber is consumed by gut microbiota, okay? And this is occurring in anaerobic conditions. So it's kind of a fermentation process. So while this fermentation process is going on in your gut, basically large intestine, it produces a lot of metabolites. Okay, it's secreting because the enzymes, the bacteria, how do they digest the food? They release all the enzymes necessary for digesting the food component. And after digestion, they take up the cells. They basically absorb into the cell, right? So while this digestion is happening, it produces a lot of metabolites. Of them, the major ones are short-chain fatty acids. And there are other metabolites. I'll be talking about maybe indole 3-propenic acid and methoxyindole acetic acid. There are many there, okay? Slowly, every day, you'll see a new metabolite being identified, which have health-promoting effect. These metabolites, they calm down inflammation. Okay, how do they do? They are released or secreted in millimolar concentration, and they're released into blood, and the blood will carry it to every organ of your body. It could be your liver, it could be your pancreas, it could be your heart, lung, brain. So the gut maintains a access with every other peripheral organ through this gut microbial metabolites. That's why we call gut-lung axis, gut-brain axis. All the axes are are maintained by these metabolites. And what do they do when they reach to the target organ? They inhibit inflammation. Why? How? How? They are a very good SDAC inhibitors. They inhibit SDAC, which is one of the histone dehydrogenase enzyme. And if you inhibit that, it prevents inflammation. It also increases T regulatory cells. T regulatory cells are T lymphocytes that produce IL-10 and calm down inflammation. Very important to calm down. Like, for example, in autoimmune disease condition, Tregs are very, very less. Okay, so in those conditions, it will be very good. They also act like an immunomodulator. What does it mean, immunomodulator? It means they are not only inhibiting inflammation, or rather controlling inflammation caused in response to any risk factor. It could be a smoke, it could be air polluter, it could be pathogen. They also improve your immunity against killing the pathogen also. So immunomodulators are not immunosuppressive drugs. They are controlling inflammation such that you will have optimal killing ability to kill your microbes, at the same time regulating inflammation, not going hyperactivated phenotype. And also there are few papers showing that it can also NR2 activate it. So the metabolites are very rich. We don't know what exactly how many are there, but it's a rich, hundreds of them are there. But most of the people have worked on short chain fatty acids, which are butyrate, acetate, propionate. Okay. Now, this is just a paper showing that lactic acid bacilli can produce this 5-methoxyindolestic acid, which activates NRF2 and prevents fatty liver. Just to show you example. Okay. Now, because a gut. Okay. Before I go, if the short chain fatty acid producing bacteria are less in your gut microbiota, then you are predisposed to every non-chronic diseases, every non-chronic disease, non-communicable diseases. Sorry. So that phenotype where you have less of short chain fatty acid producing bacteria, we call this gut microbiome dysbiosis. So gut microbiota dysbiosis or microbiome dysbiosis is associated with every disease condition. It could be autism, it could be COPD, it could be cardiovascular disease, it could be diabetes. So every disease has been shown. You have dysbiosis, there is increased risk for this disorder. And by giving these metabolites, basically short chain fatty acids, they were able to prove that it can mitigate the disease condition if it is given from very early stage. Even in some papers, they're shown to be treating the disease condition itself. So definitely, the gut microbial metabolites are good therapeutic targets. Now, because they are good therapeutic targets, people are targeting gut microbiota for prevention of disease, prevention and treatment of disease. And how are they doing it? They are using either probiotics. Probiotics are bacteria, live bacteria. They are beneficial for health. They promotes health benefits are there. Prebiotics are food for bacteria. Basically, these are the dietary fibers. Okay, dietary fiber is undigestible food component in your food, which is consumed by your bacteria. So prebiotics basically refers to dietary fiber. When you combine these two, prebiotic and probiotic, it becomes a symbiotic product. That means a fermented product. Fermentation, it could be yogurt. It's having a bacteria, as well as it's being, it's growing, it's producing microbial metabolites, and it also has prebiotics. So this kind of product is called a symbiotic. Now, some people worry that taking live bacteria. So instead of taking a symbiotic product, if you inactivate the live bacteria, that becomes a postbiotic. Basically, it's a postbiotic is nothing but a symbiotic product with no live bacteria. And how do you kill the bacteria? Either by thermal, basically heating it, or by lysing the bacteria. So both ways, people have come up with a postbiotic solution. So using this, people are now trying different clinical trials, preclinical data to show that, yes, this approach is feasible to prevent disease progression. The common prebiotics are fructooligosaccharides, galactooligosaccharides, inulin. Common probiotics, these are the approved ones. This is plantarum, lactobacillus plantarum, rhamnus, casei. Symbiotic, basically you add these two as a fermented product, but in the market, what you see is different. This is in the market. If you go and do Amazon, if you buy any, if you just put a prebiotic keyword, you will see hundreds of prebiotic coming up, and they basically are made up of inulin, fructooligosaccharides, or galactooligosaccharides, and they are marketed for promoting bowel health and regularity, reduced cholesterol, blood sugar, all that. Now, this is a probiotic. where even every big pharma is selling now. Dr. Reddy's lab, Cipla is selling. So they sell capsules containing a bacteria, which could be lactic acid, lactobacillus species or bifidobacter species, and each capsule contains 30 billion. But so far, there are no good reports suggesting that if you take probiotic capsules, improves your health. Very small number of studies are there with small number of patients where they've shown some beneficial, but not has been so promising. The reason is very unlikely that whatever probiotic you take will go on colonize in your gut bacteria, in your gut. The reason is the gut is already having a hostile environment, either because your diet lacks dietary fiber. So if there's no dietary fiber, this bacteria will not, colonize and grow. So there is a limitation of any beneficial effects caused by this probiotic. Whatever effect people are reporting could be that this bacterial population, when you take, because you're talking about 30 billion bacteria, when you take, they probably undergo lysis, and this bacterial lysis could be promoting an immunomodulating property. So there is data on that. A good symbiotic product, people are selling, basically they're combining pre and probiotic. But actually, in my definition, they are not symbiotic product. Symbiotic would be this product, which is Yakult, which a lot of people have, you all might have tasted this also. Otherwise, a lot of companies are now combining probiotic with prebiotic and calling it as symbiotic. Similarly, there are recently probiotics have been introduced to market, and they're being given for improving immunity against viral infections. And this is a product which people are recommending it for babies against viral infection. And this is a good study which I wanted to spend little time. So I told you that if you consume high fiber diet, the gut microbiota will be much more healthy in terms of its diversity, richness. Okay, so this particular group from Stanford, very well-known group, Sonnenberg, and there are a lot of YouTubes if you go and hear to them. And what they did is they took healthy subjects, divided into two groups. One group was asked to eat only high fiber diet. The other group was asked to eat more high fermented foods. Okay, and this group was asked not to eat any ultra processed food, and they could track the food log, and they could see that these subjects were complying with their instructions. And the common high fiber foods what they were having was fruits, vegetables, legumes, whole grains, nuts, seeds. And whereas the fermented foods were yogurt, kefir, fermented cottage cheese, fermented vegetables, vegetable brine drinks. So all this were rich in this particular group. And then they did every analysis in this 21, lipid profile, body weight, gut microbiome, diversity, everything they did. And they found that people who were consuming high fiber diet, there was an increase in short chain fatty acids for sure. But there was a differential responses. That means there were some patients were rather not patients. Some subjects were showing pro-inflammatory phenotype. Others were showing anti-inflammatory phenotype, and they could find out that these individuals who were showing the pro-inflammatory phenotype. That means they were higher levels of cytokines were there. They had gut dysbiosis, more gut dysbiosis compared to people who were showing beneficial effects. But on the other hand, in this arm, high fermented food diet, all people were showing good effect. That means when they looked at the cytokine levels, the mean cytokine levels of all this profile was significantly reduced from the baseline. So suggesting that, yes, fermented foods and to some extent even high fiber diet can improve your health by reducing baseline inflammation. Remember, inflammation is the driver for all the disease. Every patient has some low-grade inflammation that becomes higher and higher as the disease progresses. So if you can control the inflammation, you can slow down the disease progression. Okay. So now with this idea, we also thought that how about we make a dietary fiber rich foods, which could be a symbiotic or a postbiotic. So in our lab, what we did is we selected this millets. Why? Because this contains highest amount of fiber, ranges from 6 to 10 grams per 100 gram of Sridhanya millets. And then my students developed a process such a way that if you add the probiotic, which would be KC, plantain, brahmanas, they grow very, very well. If you just take the millet powder and add the bacteria, it will not grow. But we have developed a process that can help to grow the bacteria. I'm not going to reveal how it is done because still we are in the process of filing the patent. And then we made a symbiotic product, which is the fermented product. And also we made a postbiotic. Basically, we killed the bacteria either by lysing or by autoclaving it, basically killing the by heat treatment. And now we are working on these two products to see if we can help in reducing disease state by measuring different conditions. Okay. Now this is one experiment where we gave kodo millet postbiotics to mice which were challenged with lipopolysaccharide, which induces lung inflammation, and we found that the fermented product was sufficient. I mean, the postbiotic was very good in inhibiting the lung inflammation in this mouse. So you're comparing the LPS, this fermented, this is the unfermented. And we see that, this is just without any treatment. We see compared to LPS, by giving fermented, we could reduce the number of cells and even the cytokines was there. This is one of the first experiment. Later, we found that even unfermented product, which is basically this prebiotic, was also, quite efficient or quite efficacious in reducing inflammation. I'm not able to show the data to you right now, but you take my word, it is also effective in reducing inflammation. Okay, that's the part about the dietary fiber. Last, I'm going to stop here, maybe another 5 minutes. I want to convey this information that, we always rely on supplements, vitamin supplements compared to a natural food. So one of this project aim was to compare the antioxidant activity of vitamin C to fruit, okay, in healthy subjects. So we developed an assay to measure the antioxidant activity of antioxidants. Basically, it measures the total antioxidant capacity of the blood. How does it do? It measures the resistance of the plasma to inhibit lipid peroxidation induced by free radicals, oxidants. Basically, you're measuring the resilience of the blood to free radical injury. So how do? We take the serum from the patient sample or a healthy subject. To that serum, you add a free radical generator, AAPH, or a dye called as BODP-C11. This is a lipid peroxidation reporter dye. That means it binds to lipid peroxyl radicals and it measures lipid peroxidation. AAPH will thermally undergo decomposition and produce free radicals. So in the serum, you'll have the saturated, unsaturated lipids. Unsaturated lipids are prone for oxidation, and we can measure the lipid peroxidation in the serum. And using this fluorescence measurement, we could develop and show that this assay is very reliant for measuring antioxidant activity of, it could be a basal antioxidant, even antioxidant of any external agent, what we do, that could be vitamin C or a fruit. Here in this, what we do is we compare the vitamin C activity against vitamin C rich guava fruit. And if you look at literature, one guava fruit contains around 80 milligrams. It depends upon the variety, from 50 to 80 milligrams of vitamin C is there. So we did this study in a healthy volunteers where we gave this individual's vitamin C, which was 500 milligrams of vitamin C. And for the same subjects, we gave 3 guava fruits, which is almost coming to 200 or 250 milligrams of vitamin C, just vitamin C. But remember, vitamin C is a single molecule, but guava fruit, though it contains vitamin C, it contains mixture of many phytochemicals. If you recall from the first slide, I told you, unlike single vitamin, a complex of phytochemicals will do a better job in exerting antioxidant, anti-inflammatory, right? So now we did this study where we measured the total antioxidant capacity before and after vitamin C. We can see before, this is the mean level of total antioxidant capacity. By giving vitamin C 500 milligrams, we could see there is a significant increase in the mean and total antioxidant capacity. That means vitamin C is working. So this was done 2 hours after vitamin C tablet was taken. And so since there is more total antioxidant capacity, the lipid peroxidation also came down. So suggesting that increase in total antioxidant capacity in your serum reduce lipid peroxidation, which is an indicator of oxidative stress. So in simple language, by giving vitamin C, we could reduce the oxidative stress state in the subject. Now, how about guava fruit? Guava fruit also showed better than vitamin C. Most of the subjects who participated, they all show very good there was increase in the total antioxidant capacity by taking guava fruit. And accordingly, we saw a decrease in the lipid peroxidation. So the point I'm trying to make is consuming fruits is better than taking supplements of vitamins, because fruit contains not one vitamin C, it contains hundreds of unknown chemicals as well as known chemicals, which will be much more effective in reducing your oxidative stress inflammation than vitamin C. Okay, so with that, I want to conclude it. What I showed you is that functional foods rich in phytochemicals could be activating multiple pathways. What I showed you was an NR2 pathway as example. Besides activating an NR2 pathway, these phytochemicals also have a scavenging property. That means they can scavenge free radicals. They can scavenge hydrogen peroxide. They can scavenge hydroperoxides. And therefore, they'll be much more effective in reducing your oxidative stress. And therefore, it can reduce, slow, or even halt the progression of diseases. I also talked about fermented foods or consumption of fiber-rich foods. They all can modulate your gut microbiota and produce healthy short-chain fatty acid molecules, rather health promoting short-chain fatty acid molecules, and prevent inflammation, which drives the inflammatory diseases. Okay, so with that, I'm going out of time, I guess. So with that, I'm going to stop. I acknowledge my team. Dr. Sopanika, she graduated last year. She was the one who worked on Moringa. Deepashri and Prerna are working on the functional foods. Veena is working on piper beetle, and the other these are my collaborators. Okay, with that, I want to thank all for patiently listening to me, and if there are any questions, I'll be very glad to answer it. Thank you very much.
Good afternoon, sir.
Good afternoon.
I have a question here.
Yeah.
How to differentiate the inflammations which are due to the oxidation stress, or whether they are normal inflammations like mosquito bite, internal?
Right. So the disease could be triggered either oxidative stress as a primary mechanism or inflammation as a primary mechanism. But the consequences of increased oxidative stress or inflammation leads to both. It increase oxidative stress because these inflammatory cells, they produce free radicals. Now, depending upon the risk factor, if it is an infection, that means inflammation is a trigger. If it is a smoking, if it is air pollution, that means probably it's an oxidative stress which is driving the inflammation. So depending upon the risk factor, what you are encountering it, you can differentiate who will be the primary driver of the disease. Is it oxidative stress or inflammation?
Another question, so if the antioxidants, they are more in our body, is there any effect for that?
There's nothing called more in the body. If you are eating food, you have a limitation on food. Only if you are supplementing by taking vitamin tablets, vitamin E, vitamin C. If you have excess of vitamin C, vitamin, yeah, it has shown to be having a detrimental effect also. But when you are eating in the form of food, you know how much to consume the food, right? It's a part of the food. It's not supplement of the. So I don't see a scenario where if you consume as a food the antioxidant in the form of food, there'll be no excess as such. But if you take supplements, for sure, there are studies which can cause side effects. For example, vitamin C. Vitamin C, depending upon the levels, it can act like a good guy or a bad guy. For example, vitamin C is given for treatment of cancer because vitamin C can promote oxidative stress inside the cell depending upon the concentration of vitamin C. Similarly, vitamin E also. So it all is context dependent. We cannot have a blanket statement whether it's good or bad.
So another question, sir. According to Ayurveda, the people are having